Physiology of synapses and nerves. Synapses are the contacts between the excitable cells, between excitable structures, where excitation may pass from one cell to another cell. So, therefore, we may distinguish the synapses that may be called neuromuscular synapses between the nerve and the muscle. Here is the neuromuscular synapse. Or we may observe the synapse between one neuron and another neuron. For example, between one neuron, axon, and another neuron dendrite, exodendritic synapse, or between an axon and an axon of another cell, between an axon of one neuron and an axon of another, or between an axon and the soma of another neuron, exosomatic synapse here, for example, etc. The synapse consists, every synapse consists of three parts. We will draw the synapse. So, first part is presynaptic membrane. First is presynaptic membrane. This is an axon. So myelin sheet is finished here, and an axon produces the terminal, which inside contains the vesicles with chemical compound that is called neurotransmitter. Neurotransmitter. Inside here is neurotransmitter or mediator. So, first part of synapse is presynaptic membrane. Then we have synaptic cleft. The space between the presynaptic membrane and the postsynaptic membrane, which is a part of a second cell, for example, second neuron. This is the postsynaptic membrane, third component of synapse, and on the postsynaptic membrane there are receptors, specific proteins, and neurotransmitter may bind to these specific receptors. So we discussed the structure of the synapse, and now we will speak about the mechanism of the synaptic transmission. To the presynaptic membrane comes the action potential. So action potential excites the presynaptic membrane. So action potential propagates along the nerve fiber and reaches the presynaptic membrane, and here it induces the activation of voltage-gated calcium channels. So calcium moves into the cytoplasm and the calcium concentration increases. Calcium initiates the movement of vesicles, and they reach the presynaptic membrane. They come closer, and then they secrete the neurotransmitter into the synaptic cleft. This is called exocytosis of neurotransmitter. So the molecules of neurotransmitter, they move in the synaptic cleft, and at the end they reach the receptors, and they specifically bind to the receptors of the postsynaptic membrane. What happens then? Then the situation depends on the type of neurotransmitter and the type of its effect in the synaptic cleft and on the postsynaptic membrane. The effect may be excitatory or inhibitory, and we have classical neurotransmitters. We have classical excitatory and inhibitory neurotransmitters. Excitatory neurotransmitters, the most widespread, the most common, usual excitatory neurotransmitter in the human brain is glutamate. But also the well-known excitatory transmitters are norepinephrine, or it is called noradrenaline, serotonin, dopamine, acetylcholine may be used in the organism as excitatory neurotransmitter. Acetylcholine. They are classical neurotransmitters. And inhibitory neurotransmitters. Inhibitory neurotransmitters. In the brain, it's GABA, gamma-aminobutyric acid is most common inhibitory neurotransmitter. In the spinal cord, glycine, inhibitory neurotransmitter. So what is the difference in their effect upon the postsynaptic membrane? So if in the synaptic cleft released neurotransmitter with excitatory effect, then it binds to the receptors and opens chemically controlled channels will be induced on the postsynaptic cell, for example on the neuron, and will be conducted on the second cell, so it will pass the synaptic contact. So it's clear. And we also know that the EPSP looks like local response. And we know that local response or pre-spike is characterized by the increased excitability, by exaltation. And in that case, excitability of the postsynaptic neuron or muscle cell will increase. And just another situation occurs if inhibitory neurotransmitter is released into the synaptic cleft. In that case, sodium channels do not activate, but instead of them, potassium and chlorine channels open. On the postsynaptic membrane, potassium efflux, so potassium goes out, and chlorine, in accordance with concentration gradient, chlorine ions go in. In that case, the membrane not depolarizes but hyperpolarizes. So we can draw the inhibitory postsynaptic potential, or IPSP. Inhibitory postsynaptic potential. In that case, we have hyperpolarization of the membrane. Chlorine is negative, goes in, potassium positive goes out, so the potential decreases. We know that during the hyperpolarization, the excitability of the cell decreases. We have subnormal excitability. That's why the postsynaptic membrane and postsynaptic cell excites not so easy. The excitability decreases, so we have inhibitory effect. So we discussed the mechanism of the synaptic transmission, and now we will name certain properties of the synapses. So the properties of the synapses. The first property is one-way conduction. So of course there is one-way conduction, because neurotransmitter vesicles are contained only in the presynaptic nerve ending. Second property, high fatigability of the synapses. It's also clear because the neurotransmitter storage is limited and will, if the synapse will work constantly, decrease and synapse will get fatigued. Third property is the synaptic delay. Synaptic delay is the time that is needed to produce the synaptic transmission, and it is explained by the fact that neurotransmitter should be released, it should diffuse, it should bind the receptors, should initiate channels. It takes time. From here we have another property, low lability of the synapse, of the synaptic transmission, low lability. So lability is measured by the highest frequency that the structure, nerve, muscle, or synapse, for example, may transmit without transformation of the stimulation rhythm. So the lability of synapses is not high in comparison with nerves and striated muscles. So low lability. Another property of the synapses is the plasticity, synaptic plasticity. It means that these synapses they may change their ability to transmit excitation, and it may be the one of mechanisms of memory. There is so-called long-term potentiation. So if the synapse works, for example, the person learns to ride the skateboard, so his synapses in new areas of the brain begin to work, and their transmission becomes more rapid, and this easy transmission in the synapses stays for a long time. So a person may not ride the skateboard for a winter, but after the winter he begins to ride it well after the break, because his synapses contain this memory. So long-term potentiation is long-term change in the synaptic transmission that lay in the background of a certain types of memory, and there may be long-term depression also in some synapses. So it is quite complicated thing. Summation is another property of synapses. What does it mean? It means that if the synapse located close to another synapse, and in both synapses there is release of the excitatory neurotransmitter, then if we have sub-threshold stimulation in one synapse and sub-threshold stimulation in another synapse, there may be spatial summation, and the action potential will still be reached. The critical level will be reached, and action potential will occur on the postsynaptic membrane. This is a spatial summation, maybe also temporal summation, when the time between two stimulations is small, then one local response and another local response, one EPSP and another EPSP may occur close to each other in time, and action potential also will occur in that time, in that case. Okay, now we should discuss what happens with the neurotransmitter after the synaptic transmission, after it binds to receptor. Synapse is not single-use only structure. It may work many times, so somehow the neurotransmitter should be inactivated in the synaptic cleft on the receptors. We should distinguish three types of inactivation. The first mechanism is the reuptake of neurotransmitter. So after neurotransmitter binds to receptor and produces an effect, it may go back into the presynaptic, through the presynaptic cleft by endocytosis, and again form the vesicles or get into the vesicles that exist. This is called reuptake. Reuptake. We have many drugs that influence the reuptake of neurotransmitters. For example, reuptake of serotonin, the neurotransmitter that is related to the mood of the human or in animals. Reuptake of serotonin is blocked by the drugs that may serve as antidepressants that are used for patients with depression, because serotonin concentration in that case in the synaptic cleft increases. So one first way of inactivation is reuptake. Second, the neurotransmitter may be broken down in the synaptic cleft, inactivated by certain enzymes. For example, acetylcholine is broken down by acetylcholine esterase. This is the special enzyme, acetylcholine esterase, enzyme that breaks down acetylcholine. Norepinephrine is broken down by two enzymes, monoamine oxidase, MAO, or another enzyme, is COMT, catechol orthomethyltransferase. So you should remember both MAO, and COMT, which break down norepinephrine. For example, acetylcholine is broken down by acetylcholinesterase into the choline and acetic acid, which then may be reuptaked or inactivated by other ways. So second way of inactivation. And the third way is spillover. Spillover is when the enzyme is washed away from the synaptic cleft, eliminated and inactivated. By the way, this neurotransmitter may influence the other cells in the neighborhood, but it's another story. So we discussed 3 types of neurotransmitter inactivation in the synaptic cleft. The next question that we will discuss is the propagation of excitation in the nerves and in the nerve fibers. Each nerve consists of many nerve fibers. Inside the nerve we have many nerve fibers. So what is the nerve fiber? The nerve fiber is the process of the separate nerve cells. So for example, we have the neuron that produces an axon, and this axon in the nerve under the common shed produces the nerve fiber. So we have many nerve fibers in each nerve. The nerve fibers may be classified in accordance with the presence or absence of a special substance that is called myelin. Therefore, we have myelinated and unmyelinated nerve fibers. Myelin is lipid structure, is substance with lipid structure, and myelin covers the nerve fibers, but not completely. Like in the necklace, it produces the segments covered with myelin, and there are segments or parts of the nerve fiber that are free of myelin, which are called the nodes of Ranvier. Nodes of Ranvier, or simply Ranvier nodes. Ranvier nodes. The mechanism of excitation conduction in myelinated nerve fibers is different from unmyelinated, because action potential may only occur in the free spaces or in Ranvier nodes, because in these parts of nerve fiber the membrane is not covered with hydrophobic non-polar lipid structure of myelin. Hydrophobic myelin repels the water and ions. Therefore, only in Ranvier nodes AP may be induced. As soon as action potential due to the opening of sodium channels occurs in the node of Ranvier, it changes the potential in the neighbor nodes of Ranvier. And when critical level of depolarization will be reached in the next Ranvier node, then AP will immediately occur there. So action potential jumps in that type of nerve fiber from one Ranvier node to another, and that's why the velocity of excitation conduction is many times higher in myelinated nerve fibers in comparison with unmyelinated, where the action potential gradually propagates from one point to another along the nerve fiber, not jumping. Such jumping type of impulse conduction that may be found in the myelinated nerve fiber is also called saltatory type of conduction or jumping type of conduction. In accordance with classification that was proposed in the 20th century by Gesser and Erlanger, two physiologists, all nerve fibers may be type A fibers are thick myelinated fibers. The velocity in these fibers is 120 meters per second. Another type, nerve fibers type B. Thin myelinated fibers, they are thinner, the spaces between Ranvier nodes is less and the velocity is less, about 25-30 meters per second. And another type, nerve fibers type C, unmyelinated fibers. In that type of fibers, the velocity is about only 1 meter per second. In the body we may find all three types of nerve fibers, but in the somatic nervous system that controls the contraction of striated muscles, we have nerve fibers type A. For control of internal organs, in the autonomic nervous system, we have nerve fibers B and C, because the control of internal organs do not need so rapid conduction as in the control of the somatic nerve, in the somatic musculature. Gasser and Erlanger, they registered the action potential of the nerve. They stimulated the nerve at one part, so all nerve fibers were involved in the excitation, and excitation propagated along the whole nerve trunk. And when they measured the action potential apart from the place that stimulation was given, they get a picture that reflects the action potential of the nerve trunk. So the action potential looked like that. Here we have nerve fibers type A, and we have A alpha, A beta, gamma and delta fibers. We have here nerve fibers type B and nerve fibers type C, the nerve fibers that are unmyelinated. So action potential of the nerve looks like that. It is splitted and it is registered in the nerve trunk. You should know certain laws of impulses propagation in the nerves. We have several laws. The first law is called the law of isolated propagation. It means that in each nerve fiber the excitation propagates in isolated manner, and excitation doesn't jump from one nerve fiber to another. It doesn't happen normally. Second law is the law of two-way conduction law. It means that from the place that excitation was given, the action potential moves in both directions. So let's illustrate it on our myelin nerve fiber. In that case, the action potential will go not only to the right side, but also it will go to the left, and it will propagate in both ends, to the both parts, both ends of the nerve fiber. And the third law is called the law of physiological and anatomical integrity of the nerve. It means that if you will change the functional conditions, for example, you will apply an ice cold temperature on the nerve, the propagation will stop, will not be possible. Or you may use local anesthetics, for example, novocaine, that will block sodium channels on the membrane of the nerve fiber, and in that case the excitation conduction will also stop. It will also stop the excitation conduction from pain receptors, and that's why the dentist may work in the oral cavity and the patient will not suffer from the pain.